Hi guys, welcome into the fifth lecture of bacteriology series. Now, in this lecture, we are going to see about bacterial growth. Then we will discuss about oxygen relationship, then effect of temperature, effect of pH, and effect of salt. First of all, we will discuss about oxygen relationship. So oxygen का impact bacterial growth पे कैसे होता है? Then effect of temperature, temperature का impact bacterial growth पे क्या होता है? Likewise, we will discuss about effect of pH and effect of salt. So all we discuss all the things one by one. So without any delay, let's get started. Bacterial growth. Bacterial growth is influenced by environmental condition, three environmental condition, like three or four, you can say, presence or absence of oxygen. And then second one is temperature and pH and salt. जैसे कि मैंने आपको बताया था यहाँ पे प्रेजेंस और absence ऑफ ऑक्सीजन मीन्स ऑक्सीजन कम या ज्यादा है, उस पर डिपेंड करता है कि बैक्टीरिया का ग्रोथ कैसे होगा। टेम्परेचर हमें पता है, कुछ टेम्परेचर तो अफेक्ट करता ही करता है, बट एज इफ यू टॉक अबाउट स्पेशली अबाउट बैक्टीरिया, उस पर भी अफेक्ट करता है टेम्परेचर। एट हाई टेम्परेचर बैक्टीरिया क्या करते हैं, कैसे बिहेव करते हैं, देन एट लो टेम्परेचर कैसे होता है। लाइकवाइज वी विल डिस्कस ऑल द थिंग्स विद द हेल्प ऑफ ग्राफ एंड चार्ट। एंड देन थर्ड अबाउट पीएच, पीएच से क्या होता है इनबैलेंस, और पीएच से इम्पैक्ट क्या होता है बैक्टीरियल ग्रोथ पे। एंड साल्ट पे, हम साल्ट क्यों यूज करते हैं फिश प्रिजर्वेशन के लिए। ऑल दीज थिंग्स वी विल डिस्कस वन बाय वन इन दिस बैक्टीरियल ग्रोथ, इन दिस बैक्टीरियल ग्रोथ सीरीज ऑफ लेक्चर, not bacteriology of series of lecture. So oxygen relationship, principal gases affect growth. The two main gases which impact the growth of the bacteria. First one is, you can see, first one is oxygen, and second one is carbon dioxide. This first one is oxygen and second one is carbon dioxide. So principal gases affect the growth is the types of bacteria based upon their relationship of oxygen. So principal of gases is oxygen and carbon dioxide, but we are going to see mainly about oxygen because oxygen is most important. What oxygen can impact on the growth of the bacteria. So types of bacteria based upon their relationship to oxygen. So first of all, I will talk about aerobic bacteria, or we can call them as aerobes also, which require molecular oxygen for growth. These bacteria, aerobic bacteria, require molecular oxygen. You can say free oxygen required for their growth. They require oxygen, O2, for their growth. Then if we talk about anaerobic bacteria, we can also call them as anaerobes, which are unable to grow in presence of molecular oxygen. Uh, in O2 presence, they does not grow. Like you can see, this is oxygen molecular bonding structure. So in this presence of oxygen, aerobic bacteria can survive, but anaerobic bacteria can't survive in presence of oxygen. So doesn't need molecular oxygen for their growth, and they can survive without oxygen, like carbon dioxide and all. Like facultative anaerobes, facultative anaerobes which can grow both in presence of absence of molecular oxygen. They grow in presence of oxygen or absence of oxygen. इनको घंटा फर्क नहीं पड़ने वाला, ऑक्सीजन हो या ना हो, there is no impact on their growth. Then macro aerophils which which will grow under oxygen tension less than that of the air. If we talk about macro aerophils, means they need oxygen up to some optimum level, means जो नॉर्मल कंडीशन में ऑक्सीजन होता है, अंडर नॉर्मल टेंशन होता है, उससे कम होना चाहिए। इन अदर वर्ड्स में बात करना चाहूँ तो ऑक्सीजन लो अमाउंट में होना चाहिए। इन सिंपल लैंग्वेज, ऑक्सीजन शुड बी लो इन मैक्रो एरोफिल्स कंडीशन. So this is all about oxygen relationship. Relationship about this is all about oxygen relationship. Principle of gases we had studied in this slide: oxygen, carbon dioxide impact. Like that, especially we discuss about oxygen, what is the oxygen can impact on aerobic bacteria, anaerobic bacteria, facultative anaerobes, and likewise we study about macroaerophils. So next we will discuss about next part of this, effect of temperature. So in this thermometer, you can say the temperature is can be seen. So influenced by temperature, each species of bacteria grow at temperature within certain temperature, certain or certain temperature is typing error here. Each species of bacteria grow at temperature within certain limits, you can say. Every bacteria need kind of some or optimum level of temperature. They influenced by bacterial growth. Temperature influenced by bacterial growth. So with reference to the temperature requirement for their growth, bacteria are commonly classified into three broad groups. with reference to temperature. We are considering as a temperature basis of classification, commonly classified into three. On the basis of temperature, we are classifying bacteria, like bacteria growth in three format: sacrophiles, mesophiles, and thermophiles. So discuss about first, we will discuss about all these things: sacrophiles, mesophiles, and thermophiles in the help of diagram. So let's see. This is a graph, you can say. This is sacrophiles, mesophiles, and thermophiles. So especially this is designed with the help of K. Bhalchandran book, specially because all the things are which are given in this chart are exactly taken from this book. So no need to worry. So sacrophiles. Sacrophiles are cold living. As name suggests, they are cold living. Means they love coldness. Okay. So sacrophiles are cold living. Then 0 degree Celsius to 10 to 20 degree Celsius temperature they need to grow. Then 0 degree Celsius to 20 degree. Optimum they need optimum growth temperature about 15 degree Celsius. Optimum is specially which in which they peak growth is in. ऑप्टिमल टेम्परेचर मतलब कुछ भी नहीं कि जहाँ पे उनको अच्छे से ग्रोथ दिखती है, ऑप्टिमम कंडीशन वाली. कैपेबल ऑफ स्पॉइलेज ऑफ फिश प्रिजर्व एट लो टेम्परेचर. अगर हम सोचते हैं कि फिश को हमने आइस में रख दिया तो फिश स्पॉइलेज नहीं होगा, दैट्स ट्रू, बट it's not completely true. Because some bacteria are cold living, they are also present at survive at the ice level, but not that minus degree Celsius, because 0 degrees Celsius to 20 degrees Celsius is a kind of cold, you can say, by our point of view. Bacteria associated with spoilage are Pseudomonas, Achromobacter, and Flavibacterium. This is the important point here because by JRF point of view. In exam, the question will be like this: which are the sacrophiles bacteria? So Pseudomonas, Achromobacter, and Flavibacterium, and third one will be other, and odd one out are likewise. Question will be there. So this temperature and this degree Celsius and optimum, this is the most important example, just keep in this mind. Mesophils bacteria medium temperature show maximum growth between 20 and 45 degree. The bacteria which show temperature between bacteria, the temperature growth in between 20 to 45 degrees Celsius, you can say. Maximally this kind of temperature will be in rainy season in India. So medium temperature is 20 and 45 degrees Celsius. Optimum temperature growth lies between 30 and 37 degree Celsius. So ज़्यादा का जो peak growth होती है कब होती है? 30 and 37 डिग्री सेल्सियस के अंदर होते हैं. डू नॉट बिलो 5 डिग्री सेल्सियस. 5 डिग्री सेल्सियस के नीचे चला गया तो इस बैक्टीरिया की ग्रोथ कंप्लीटली स्टॉप हो जाएगी. मीसोफिल्स बैक्टीरिया. मीसो मीन्स बीच वाले. फाइल्स मीन्स मीसोफाइल्स मीन्स फाइल्स मीन्स समथिंग लाइक दैट, लविंग और लाइक दैट. Hydrophiles, hydrophilic, like that. Phils means loving someone, like that. Mesophils means they love medium temperature. Thermophils means temperature, high temperature they love, like that. Optimum temperature growth lies between 30 and 37 degrees Celsius. Do not develop below 5 degrees Celsius. Disease causing bacteria. These are the disease causing bacteria, especially mesophils. Consist of terrestrial flora like Bacillus and big typus, Carinoforms, and Macrococcus. These are the example of these mesophils. Just one thing remember in this point, important is that is mesophils cause any diseases? Does mesophil cause any disease? Like his example is that disease causing bacteria are mesophils, especially keep in this mind. So the most important by JRA point of view is in also is that temperature, what is the range of temperature and degree, like 20 to 45, and optimum temperature growth is 30 and 37 degrees Celsius, and likewise below 5 degrees Celsius does not survive, and disease causing bacteria consists of example. Just remember this, which are the example of mesophil bacteria, like that. Then you have to quotes here that Corionoformus and Macrococcus. The Macrococcus, these are the example of this bacteria. Then if you talk about thermophiles, thermo means temperature and philes means something loving, like that. I already told you. High temperature. Thermophiles means high temperature. The bacteria which are survive at a high temperature, these are the thermophiles. grow in between 45 and 65 degrees Celsius. This bacteria specifically grow in between 45 and 65 degrees Celsius. Optimum temperature is 55 degrees Celsius. The peak growth of this bacteria is in up to, about 55 degrees Celsius. Survive in hot spring temperature 85 to 90 degrees Celsius. The example of such bacteria are lactobacilli, streptococcus, and thermophiles. Yeah, we are familiar with the lactobacillus because this lactobacilli, these bacteria are used for fermentation process specifically. Then this is about saccharophiles, mesophiles, and thermophiles. Just remember in this chart, just take a screenshot of this chart and mug up this because it's really, really important by JRF point of view. Yeah. And now we will move to our next slide. So this post of the technology. So this is the chart which is directly taken from post-harvest technology, fish and fish product, K. Balchandran. So saccharophiles, the temperature is 0 to 20. Mesophiles, you can take a screenshot of this also, and you can read all this also. So this will be easy for you. So effect of pH. Effect of pH. What is the effect of pH? You can see in this graph. Sorry. In this graph, acidophiles, neutrophiles, and alkylophiles, these are the given here. That acidic means acidophile, neutral means neutral, alkaline means alkaline. We know all these things because pH is varies. So effect of pH. What effect of pH cause on the growth of the bacteria? So let's see. There are few point here. Acidic or alkaline pH are harmful or to growth of the bacteria. Acidic or alkaline. You can see 0 to 7 is acidic and 7 exactly is neutral, or 6.5 to 7.5 you can say it's neutral, but after 7.5 to 14 it will be alkaline. So for optimum condition or for growth of the bacteria, their medium should be 6.5 to 7.5. The pH beyond either in acidic or alkaline range is unfavorable. Yeah, that's is important because the range you have to keep in mind here only 6.5 to 7.5. That's nothing else. Okay. Now next we will move effect of salt. So the question you this is important here. Why we use salt for preservation of fish? So because see, when we use ice, what happened? Let me show something. Yeah, I will tell. Now this is the fish, you can say. So this is the fish. What I am doing, I'm applying salt. This is the salt, you can say. I'm applying salt on this fish totally from outside to inner side. Let me take salt color. This is a white salt. Okay, so I'm applying white salt on this fish. Now what happened? The salt, salt NaCl, what is NaCl molecule is salt. So what NaCl do? The water which are present inside the fish, which is bounded water, which is removed with the help of NaCl, Na plus Cl minus, as we know H2, H plus, that binding is there. So that water get removed from binded water, which is get removed from fish body, and the bacteria does not get chance for what we say for growth or microbial growth. So likewise, so salt role in prevention of microbial growth. Salt is effective as preservative because it reduces the water activity of foods. The water activity of food is amount of unbound water available in microbial growth and chemical reaction. Does salt inhibit bacterial growth? Yeah. Salt act as a preservative by inhibited microbial growth. Salt act by drawing water out of the cells. Yeah, I already told you. Salt, what they do when we apply salt outside the fish, the salt removes the water from that cell, and this is the due to process of osmosis from higher concentration to lower concentration. Water, the phenomena is like that. Water get like banded and NaCl removes it, reducing the amount of water available in bacteria, inhibit the slows bacteria growth and reproduction. Yeah, bacteria need water for their multiplication. Basically, we had studied about chemical composition of bacteria. 90% is the water is present in bacteria. In previous slides we had studied about that. How does salt interaction with bacteria? Due to its antibacterial properties, salt has long been used in preservatives. We knows that we use apply for in canning, like in packaging processing technology. We will study about what is impact of salt on the fish and like that. But in short, we are studying here also now because of osmosis. This process occurs. Salt kills some types of bacteria effectively by sucking water out of them. In process known as osmosis, water passes out of bacteria as balanced salt concentration each side. Yeah, this is cell membrane and all. We this is simple things. Just read out till. Yeah, I explained you. Let me clear this. Yeah, clear all drawings. Yeah, now next we will discuss about effect of salt by in as it is which is given in K.K. Balchandran. These lines are sorted here. So first of all, marine bacteria require around 0.5 to 3.0 salt. Specifically, marine bacteria required this kind of range in which bacteria can survive in marine bacteria. Many bacteria do not grow when the salt concentration is in the range of 10 to 15 degrees Celsius. So the salt which apply, its range is much more effect of salt. Halophilic bacteria develop in concentrated solution of NaCl. Halophilic means you can say salt loving also. Halophilic bacteria, concentrated solution of NaCl also, because they love NaCl, or I don't know what the is a phenomena or like that. But and by our point of view, it's not important that halophiles what their chemical composition is, why they survive in NaCl. Bacteria require a low concentration of salt for their growth. Bacteria specifically, they does not require at all salt, but require, if they require salt, it should be in low concentration. This is all about this sacrophiles, mesophiles, this is all about this salt and all, then effect of pH and effect of salt. Thank you for watching this video. For next video, go in our playlist and check out there. Thank you.